In part 3 of chapter 5, we're going to consider other kinds of catabolism than carbohydrate catabolism. Once cells run out of carbohydrates, especially glucose, then they turn to breaking down other materials and getting energy from them. So first we'll consider lipid catabolism and talk about how fats and oils, specifically triglycerides, are broken down, and then protein catabolism. And then with both of these pathways, they also have a role in anabolic reactions, so we'll mention that as well. And then later we'll turn our attention to photosynthesis, which is anabolic. So in lipid catabolism, the main lipid that I want to talk about breaking down is the triglyceride, which we covered back in chapter 2 as being 3 fatty acid chains connected to a glycerol backbone by ester bonds. So this is a polymer version of a lipid as opposed to the steroids that we had talked about previously that are not polymers. So first thing to do is we have to break the monomers, the fatty acids, off of the glycerol backbone, and that's done with an enzyme called a lipase. So lipase is, as you can see in the name, ends in ASE, so an enzyme that, and the reaction it does is that you can see that we're adding in water here. So that's a hydrolysis reaction, using water to break these covalent bonds, the ester bonds that hold on the fatty acid chains. So that hydrolyzes ester bonds to release fatty acids from glycerol. And so we can see after that reaction that we have our glycerol molecule, and then we have the 3 fatty acids, which are both dealt with differently. In the case of the glycerol molecule, that's going to be phosphorylated, and then that makes DHAP, which we had seen as an intermediate in glycolysis. DHAP was 1/2 of the glucose molecule, so this is going to get converted to G3P, and then can continue through glycolysis, the rest of glycolysis, that second half, where you can make a couple of ATP from that. Alternatively, if the cell has enough energy, then it might use that G3P, or use that DHAP to connect to a G3P and make some glucose. We're considering catabolism here though, so we'll assume that the cell needs energy and is going to break that all the way down. So then the other portion of the triglyceride is the 3 fatty acid tails, which, as we can see, are much larger, a lot more carbons involved with a lot more bonds between those carbons, which means lots of electrons that can supply some energy in breaking those bonds. So that's where the real energy comes from in fats, oils, triglycerides, is from the fatty acid tails. If you look at their composition, that it's all just carbons, and then what they haven't drawn in here is the hydrogens. So a hydrogen there, a hydrogen there, or on the internal carbons you just have a couple of hydrogens there. But there's really nothing else there other than carbon and hydrogen along the entire length of those molecules. So that makes them pretty dense in terms of the molecular weight ratio to the number of bonds that are there. And so lots of bonds to break compared to, say, a glucose molecule. The glucose only has 6 carbons in it. It has also 6 oxygens that take up some of the weight of that molecule and don't contribute any energy. towards that. The oxygens don't get broken off. So therefore, you can understand how fats and oils having 9 calories per gram because they have so much more energy in them than, say, carbohydrates that are only 4 calories per gram. So in order to break down the fatty acid components, go to the next slide here, that shows it being done by a process called beta oxidation. We're basically going to cleave off these acetyl groups in this fatty acid. So acetyl is 2 carbons. So we're just going to break off 2 carbons at a time, chewing from one end and just taking off these 2 carbon chunks until the molecule is completely broken down. Those 2 carbon chunks, or acetyl groups, get hooked onto coenzyme A, and that makes the acetyl-CoA, which we talked about before in the Krebs cycle. As this is the molecule that brings the carbons left over from pyruvate to the Krebs cycle. So now the acetyl-CoA has a different origin here. It's from a fatty acid, but it's treated just as same as the 2 carbon acetyl group from pyruvate, that it enters into the Krebs cycle and then goes around the Krebs cycle being lost, the carbon being lost as CO2, allowing you to convert a bunch of electron carriers to the reduced form, and then that allows you to run a bunch of electron transport chain, which gives you lots of ATP for the cell. So electron carriers reduced, and we can see both NAD and FAD in this process. So we make NADH, the reduced form, and FADH2 in this process. And those, again, go to Krebs, or to the electron transport chain, excuse me. So this is all going on in the cytoplasm of prokaryotic cells, and this happens in the mitochondria of eukaryotic cells. So these reactions are going on in the cytoplasm of the prokaryotic cell or in the mitochondria of the eukaryotic cell. So the next nutrient to consider breaking down is proteins. Proteins outside of the cell are too big to enter the cell, as there are many dozens typically of amino acids in length, maybe even hundreds of amino acids in length. And so we have to break those proteins into their individual amino acids with using a hydrolysis reaction and enzymes called proteases. So proteases ending in ASE and being something that breaks down proteins. Fairly simple to remember. Proteases are just a large class of individual enzymes that break specific bonds in proteins. Some proteases specifically break or hydrolyze the peptide bond between, say, an alanine and a tryptophan, while another specific protease with yet a different name might hydrolyze the bond between a glycine and a glutamate, for instance. So we're just going to consider them all generically as proteases. And in the picture here, we can see we have this large protein. We're going to break it up, breaking the peptide bond that exists between the amine of one amino acid and the carbonyl, which was a carboxyl group of the previous amino acid before they formed a peptide bond. So we can see the junction, the peptide bond between those. We're just going to go through and break those down with our different proteases so that then we have separate amino acids. And then those separate amino acids are brought into the cell by some kind of permeases that allow those molecules to go through the cell membrane. As soon as they get through the cell membrane, then we're going to start breaking down the actual individual amino acids by first removing that amine group from the amino acid. That comes off in a process called deamination. So deamination is just removal of an amine or an amino group from an amino acid. And then the remaining organic molecule looks like something fairly similar to those molecules in the Krebs cycle, and so then it can be sent over there to the Krebs cycle and further broken down in that cycle. So we've talked about the Embden-Meyerhof pathway, which is, or glycolysis, and that's what most cells do. But some organisms, such as Pseudomonas and Enterococcus, do what is called the Entner-Duderoff pathway. I know the names are a little bit confusing because both the Embden-Meyerhof pathway and the Entner-Duderoff pathway are named after German researchers that discovered them, but they are talking about different things. So the other pathway here is the pentose phosphate pathway, which can be used to break down glucose, but not so much for deriving energy from the glucose. It's usually used to make other molecules that can be used in anabolic reactions. So we'll talk about these individually. So in looking at catabolism of any nutrients, this nutrients over here is going to include carbohydrates, lipids, proteins, and could even be nucleic acids if they're available to the cell. That they're going to come in, we're going to break those different nutrients down, get some ATP from them, and then use that energy and those smaller building blocks to run some anabolic reactions, making different macromolecules that can make cell parts and allow cell processes to go on. And so we see that the textbook has shown catabolic reactions in purple and anabolic reactions in teal. And that same thing continues on this more complicated view of catabolic and anabolic reactions. The main point, don't get lost in the details of this figure, the main point I'm wanting to show here is just that these two kinds of reactions, either breaking down nutrients or making molecules in the cells, can be coupled. And in fact, the pathways that we've been talking about going in the catabolic reaction can be run in reverse in the anabolic reaction. And so there's a lot of reactions in the cell that are called amphibolic, meaning that they can go both directions, either catabolic or anabolic. And so when we look at some of these pathways for the different nutrients we've been talking about, we have sugars coming into the cell, such as glucose, and we've talked about how to break that down, how to run the Krebs cycle, and the electron transport chain with that glucose, and make a whole bunch of ATP, doing respiration. And we've talked about also now breaking down lipids and to their fatty acids, and then sending them over to the Krebs cycle as well. And then now I want to show you the alternate pathways. is that sometimes you take some of this glucose and instead of breaking it all the way down to CO2 and making lots of ATP from it, you may want to make some more nucleic acids for your cell. The cell is going to maybe go through binary fission and needs to copy its DNA again. You need the individual nucleotides to be able to build that copy of the DNA. And the way that you get those is by taking some of that glucose available to the cell, sending it through what is called the pentose phosphate pathway or pentose phosphate shunt, and then that's going to allow you to use that glucose only partially broken down to make the 5-carbon ribose sugar that's the base of the nucleotide. All right, so so this is going to be running. We're going to run partly catabolic with our glucose. You can see purple arrows here, but then when we go to make nucleotides, we're switching to our green arrows for that. So the pentose phosphate pathway, just think of that as an alternative to glycolysis. So an alternate pathway for glucose in the cell. Instead of going through glycolysis and respiration, we're going to break down glucose a little bit, and the main point of this is to make many precursors, and especially those that have 5 carbons or pentose sugars. For example, ribose being one of the primary ones. Ribose, and then you can make deoxyribose from that so that you can make either DNA or RNA nucleotides. So another difference between this pathway and glycolysis is that we have a different electron carrier here. Notice that in this pathway we're going to produce 2 NADPH, that's from NADP+. So we're going to add electrons onto that electron carrier, a couple of those electron carriers, and reduce it to NADPH, which we'll talk about what a possible pathway for NADPH is a little bit later on when we go through the details of photosynthesis. That's where you can use that. One place you can use that electron carrier. And then we also make a little bit of ATP. So as an alternative to glycolysis, again, the point here is not energy, because this 1 ATP, that's 1/2 of what we make in glycolysis, where you make 2 ATP. So you don't get as much energy yield here, and you still reduce a couple of electron carriers, so there's not much difference there. But the point is you get these pentose sugars, which can be made, lots of different pentose, or at least several different pentose sugars, which can be made into other useful molecules in the cell, and that saves the cell a lot of energy. If you can take a larger carbon molecule, break it down part way, and make something useful for the cell, that offers a lot of savings than having to take individual carbons, stick those all together, and use all the energy to create those bonds. Would be a way less efficient process. So many organisms use this in addition to glycolysis to build those precursor molecules they need for anabolic reactions. So then we have the Entner-Duderoff pathway. This is also an alternative to glycolysis, where you have organisms that use this rather than the Embden-Meyerhof pathway, and basically for the same reason, that they're using it to break down glucose, and they break it down to 1 NADPH and 1 ATP. And you might wonder, well, why do they do this? This is much less efficient. Well, they still have other pathways that are going to follow the products of the Embden-Meyerhof, or excuse me, the Entner-Duderoff pathway, such as anaerobic respiration to continue breaking down that glucose. And so there's not that big of a difference in the amount of ATP. There's half as much ATP from glycolysis, but then the downstream pathways are more efficient in their use of glycolysis. So it doesn't put these organisms at that significant of a disadvantage. Either way, only a few organisms do this. Enterococcus, which we work with in the lab, although we don't use this particular test, any tests for the Entner-Duderoff pathway to identify this organism. And then we also have Pseudomonas that does this. So I don't expect you to remember those, just connecting some of these pathways with some of the organisms that are familiar to you from what we've talked about in the lab. So the main take-home points that I want you to understand about this is that you're breaking down glucose, but you're making chemical intermediates for anabolic reactions, especially in the case of the pentose phosphate pathway, and then you're using it at a different electron carrier. NADPH is made rather than NADH. So make sure you understand the difference between those two electron carriers. Those are very different things. Okay, so I mentioned that we would talk about NADPH again in photosynthesis, so we'll get to that in this section of the lecture, as well as talking about lots of other details of photosynthesis. So when you talk about photosynthesis, there's really two parts of photosynthesis. There's the photophosphorylation portion at the beginning, and then there's the broader process of photosynthesis. So the photo portion of photosynthesis is referring to photophosphorylation, and then the synthesis part is going to be making sugar. Which that happens by another set of reactions, starting with something that's called the Calvin-Benson cycle and gluconeogenesis. And so we'll cover that eventually. So other things in here define photosystem, reaction center, and then I'll add to this gluconeogenesis. And that might not be represented on the study guide because not all microbiology instructors cover that, but I do want you to know what that term is, and we'll cover it a little bit later on. So in considering photosynthesis, synthesis implies that you're making something, so that's an anabolic reaction. That we're eventually going to be doing, but as I mentioned, synthesis is actually the second half of photosynthesis. We've got to talk about the photo portion of it first. So photophosphorylation is simply making ATP from light energy also known as photons, that can come from sunlight, can come from artificial light, doesn't matter. And then in doing photophosphorylation, different kinds of organisms do this. We're familiar with photosynthesis that is done by photoautotrophs. So they do photosynthesis. The synthesis being taking CO2 and making organic compounds out of that, making them a photoautotroph. But then there also is photophosphorylation that's done by photoheterotrophs, that they make ATP from sunlight, but they aren't doing the carbon fixation, the synthesis part of photosynthesis. They're just doing the energy yielding part in the beginning of that, and they actually require organic compounds made for them. So then remember that photosynthesis is photophosphorylation plus carbon fixation. That's the synthesis part. And in prokaryotes, this is going to happen in the cytoplasm and on the cell membranes, the what are called the thylakoid spaces of the cell membranes, where you have the cell membrane very highly folded upon itself to make lots of surface area for all the light capturing proteins and pigments that are needed. And then in eukaryotes, this is going to occur at the thylakoids of the chloroplast, so membranes inside of the chloroplast. And in eukaryotes, we only have photoautotrophs. When you're talking about photoheterotrophs, there are only prokaryotic organisms that can do that. So in photosynthesis, I usually think of it as being the reverse of respiration. In respiration, we started out with a sugar, and we broke that down, and in the end of that process, we ran an electron transport chain and made some ATP. But in the way that photosynthesis is the opposite of that is you're not starting out with a sugar. That's the end. Instead, you're starting out with capturing some light energy, and that allows you to run an electron transport chain, make up a proton gradient that allows you to make ATP. So you're making ATP in the beginning, and then you use that ATP and along with some electrons and moved by electron carriers to connect together a bunch of carbons to instead make sugar at the end of this process. So again, respiration, breaking down sugar to make ATP. Here we're starting out with an electron transport chain that makes ATP, and then at the end of the process, making some sugar. So in photophosphorylation, again, this is just making ATP from light. We're going to have to use pigments to do this. And so pigments differ between different organisms. Their purpose is essentially the same. We want some pigments that capture light and then start this electron transport chain. Different pigments are going to capture different wavelengths of light or different colors of light. So we have in the green plants, you're probably already familiar with, the pigment there is chlorophyll. And that same pigment is used in algae and cyanobacteria as well. And then you have bacterial chlorophyll. Which is used by green and purple bacteria. The significance of this, actually, these two different wavelengths, or excuse me, two different pigments receive different wavelengths of light in order to operate an electron transport chain. And the significance of this is that you can have the purple bacteria living in different places than algae, depending on what wavelength of energy is being received there. So to begin photophosphorylation, we first need a photosystem, which is pictured in the figure below. And so in their photosystem, it's going to consist of accessory pigments, those accessory pigments pictured out here in the periphery of this photosystem, they're working basically like antenna, that they receive this wider signal of energy that they can focus on the reaction center of our photosystem. So accessory pigments and a reaction center. To receive photons. Or light energy. And then at the center of that reaction center, you're also going to need something to receive some electrons to start the electron transport chain. So I'll say an electron acceptor. Begins the electron transport chain. So what's important to understand in this figure is that the photons, or light energy, is being collected by a bunch of accessory pigments. These are the non-chlorophyll pigments, many of them, the same pigments that you see in fall leaves when they change color, when you go to yellows and reds and even purplish colors. A lot of those are the accessory pigments that are collecting different wavelengths of light in addition to what some chlorophyll molecules in here are collecting. And then they all get passed on, the energy gets passed on to other molecules in here, passing all that energy until it gets to a central chlorophyll molecule at the reaction center. And then this chlorophyll molecule here has a couple of different oxidation states. It has electrons on it, and then those electrons are going to be bumped off by that energy that is being received at that central chlorophyll molecule. And then those electrons are going to go get bumped off to this acceptor molecule. So the acceptor there is where those electrons are going to go, and the acceptor is the start of the electron transport chain. And then they'll move down that electron transport chain, just as we've talked about before in respiration. So the electron transport chain in photophosphorylation, just like the electron transport chain in respiration, leads to forming a proton gradient, and also leads to chemiosmosis, those protons wanting to go back across that membrane. But they can't cross the membrane because they're charged. They can only cross through ATP synthase, again, as we talked about with respiration. But an ATP synthase still makes ATP in this process, but it's powered by protons that came from a different source. They came from light energy. So this is called photophosphorylation. So in cyclic photophosphorylation, the electrons that have left our reaction center, so reaction center right here, the electrons, as light energy comes in, those electrons get bounced out of that chlorophyll molecule, go on to our acceptor, and then from the acceptor through the electron transport chain. So this is all electron transport chain here. And then the electrons go back into the photosystem. So there's no external source of electrons needed. They're just making a circular path as they go from the reaction center through the electron transport chain and back. And there's more than one going on. So you can think of this as almost like a flow of electricity. You got these electrons just circulating through all of these different proteins in this between the system and the electron transport chain. And as that system runs, as that current is flowing through those, we're going to be pumping protons across that membrane, which causes that large proton gradient that makes all the ATP in photophosphorylation, at least in this portion of cyclic photophosphorylation, which cyclic photophosphorylation can be the only photophosphorylation that an organism does if you're talking about organisms that are photoheterotrophs. Now, in saying that this is the only photophosphorylation in photoheterotrophs, I don't mean to exclude photoautotrophs from being able to do this. They can do this too. It's just that in photoheterotrophs, they only do this. They don't do the next photophosphorylation we're going to talk about, of non-cyclic photophosphorylation. So what's non-cyclic about this is that the electrons don't make a return trip as they do in cyclic photophosphorylation. So that means that then we do need an external source of electrons. So our source of electrons here, you can see in this picture, is water. That we're going to actually split a water molecule, take the electrons off of those hydrogens associated with the oxygen, and pass those electrons through the photosystems. In doing that, that frees up oxygen, which makes this oxygenic photosynthesis, oxygen-generating photosynthesis. There are alternatives for organisms that are doing non-cyclic photophosphorylation, where they can take electrons from other compounds rather than water, and in the end they don't generate oxygen. They would be anoxygenic photosynthesis. All right, so back to our photophosphorylation. The electrons are not cycled, and another difference is that there are two photosystems used. Instead of just photosystem 1, as we described previously, now we have photosystem 1 and 2. They're not used in the order that they're numbered. They're numbered for the order they were discovered, which makes it a little bit confusing that these actually operate in the backwards numerical order. That we're going to start with photosystem 2 and then move electrons into photosystem 1. And this is only used by autotrophs. So photoautotrophs, those organisms that are doing photosynthesis, are the only ones that are doing this. I guess we can say photoautotrophs. Be more specific. All right. So in looking at this pathway overall, we have our electrons again coming from the water molecule, and we're making some oxygen as we talked about before. So we know this is oxygenic photosynthesis. The electrons are needing to go into this reaction center because as light comes in, we're exciting the chlorophyll and other accessory pigments in this reaction center, and then that central chlorophyll is losing electrons that are going to go to an acceptor and then down an electron transport chain. So we have our electron transport chain here. And those electrons don't come back. So then that means we need to keep breaking down water, keep splitting water molecules and taking the electrons away from those water molecules to replace the electrons that were lost from the reaction center to the nearby electron transport chain. All right, so once the electron transport chain has electrons moving through it, those electrons' destination is actually the next photosystem. Same thing going on this photosystem. We have light coming in, exciting those chlorophyll molecules, exciting that central chlorophyll molecule, having it raised, its electrons raised up to a higher energy state where they get bounced out to the electron transport chain. Our second electron transport chain here. And those electrons aren't making a return trip, so we need electrons from the first photosystem to come in here and replace those. So we're only splitting water at one location at that beginning of photosystem 2, and then those electrons travel through both of these photosystems. Where the electrons end up after the second electron transport chain is they get dumped onto NADP+. So again, a different electron carrier than the NAD+ that we've been talking about. NADP+, you can remember that this one is used here because it's got a P in it, and this is for photophosphorylation. Dump the electrons on there with a hydrogen, and that reduces it to NADPH. NADPH is going to go on to another pathway called the Calvin-Benson cycle, which is going to do the synthesis part of photosynthesis. And we're not done yet with photophosphorylation. We've talked about the photo part of it, but not the phosphorylation part. So we've got this proton gradient out here. All these protons want to get back across. Again, we talked about that energy before being proton motive force. And that they're going to move across this membrane through this enzyme called ATP synthase. And so they're doing chemiosmosis. Again, not osmosis because it's not water, it's a proton. Chemiosmosis, allowing or describing these protons coming back across the membrane through ATP synthase, and then that combines an ADP and a phosphate to make ATP. So photophosphorylation. So in summary of our photophosphorylation, we have pigments absorbing light energy. The light energy is used to create a proton gradient via an electron transport chain or two. And in cyclic photophosphorylation, you just have the one reaction center and electron transport chain, and the electrons are recycled back into that reaction center, making a return trip. In non-cyclic photophosphorylation, there's a final electron acceptor of NADP plus that gets reduced to NADPH, and therefore you need a source of electrons. It can be water in the case of oxygenic photosynthesis, or other things in anoxygenic photosynthesis. And then in either case of either cyclic or non-cyclic photophosphorylation, you have ATP synthase allowing those protons to come back through the membrane and generating some ATP in that process. So I mentioned anoxygenic photosynthesis, and that's one difference that I wanted to point out on this slide, that sometimes organisms oxidize H2S, two elemental sulfur, and you can see in H2S it looks a lot like H2O, and that you have an element that is connected to some hydrogens, and those hydrogens don't hang on to their electrons very well. We can separate the electrons from those. So then instead of it generating oxygen, you're just generating elemental sulfur. So that's an example of anoxygenic photosynthesis. Also in the photosystems that were outlined here, it's just a generic model. There's different kinds of arrangements for electron transport chains, and photosystems can be different as well when you're looking at different species of organisms. So now we come to the synthesis part of photosynthesis. So this is where we take our electron carrier, NADPH, that's in its reduced form carrying electrons, and the ATP from those light-dependent reactions that we had previously talked about, and now do a light-independent reaction. So this can happen in the dark after the sun goes down in photosynthetic organisms. It goes on during the day too, but it's not dependent on sunlight like photophosphorylation is. So we take the electrons, we take the ATP, we're going to bring in some CO2, and we're going to make some organic compounds, do some carbon fixation here. So that's what that arrow represents, carbon fixation, going from CO2 to glucose or any other organic carbon compound. So this is only done in photoautotrophs. They're the only ones that can do carbon fixation. And as I mentioned, we need some ATP, we need some NADPH, and we need some CO2. In fact, 6 CO2 to give you each of the 6 carbons in glucose. So we're going to reduce that CO2. This is highly oxidized form of carbon. We're going to want to connect those carbons together by them sharing some electrons, creating some covalent bonds between them. And so we're going to add in some electrons from our NADPH. And that's going to require some energy to do that, and that's going to come from the ATP. All right, so in reducing CO2 to glucose, we're going to do that in a couple of steps. The first step is actually a cycle called the Calvin-Benson cycle. That's going to take in 3 CO2s at a time and make it into G3P, which is, it's kind of complicated. And so we'll have to do 2 of these cycles to get enough carbon or enough G3P units to make a single glucose, which is a second part of a cycle, or a second part of this reaction. So this portion right down here is where we actually make our glucose. This is called gluconeogenesis. I'll return to that in a minute. All right, so starting out with our CO2 up here at the top of the cycle. In the cycle, we have a 5-carbon molecule called ribulose bisphosphate. And so I'll just label, I don't care if you know the name of that molecule, just remember that in the Calvin-Benson cycle, you have a 5-carbon compound. And we're wanting to add CO2 to that. Notice that they have numbers here, that you're doing 3 CO2 and connecting each one of those CO2 to one of 3 ribulose bisphosphates. So they show a single turn of these arrows going on, but you would actually have 3 of these pathways going on at the same time, like one on top of the other, so that we can take in 3 CO2s. For simplicity's sake, we'll just be following the 1. So CO2 comes in, gets connected to this 5-carbon compound, and that gives us, so this plus this, gives us a 6-carbon compound that then gets split in half. But this isn't the 2 3-carbon compounds that we're going to make glucose out of, because it's still very highly oxidized. We just connected very oxidized carbon to this. So now we need to do some more reactions. So we're going to go ahead and phosphorylate this molecule. So this molecule, phosphoglyceric acid, again, don't worry about the name. Just know that there's 3 carbon compounds here. It might help you to add up the totals of these. So you have 6 times 3 carbons equals 18 carbons. Up here we have 3 times 6 carbons equals 18 carbons. And then down here we have 3 times 5 carbons equals 15, plus 3 carbons that we're bringing in equals 18. So all of our carbons still here. We haven't lost anything, is my point with doing that. Don't worry so much about being able to do that math. I wouldn't ask you to go through that. Again, I just want you to mainly remember that there's a 5-carbon compound that's going to take on single CO2 carbon units. And that gives us our 6-carbon compound that gets split to 2 3-carbon compounds. And now we're taking these 3-carbon compounds and we're phosphorylating them. Phosphorylating them, that's going to use up some of our ATP, and then we're also going to be taking some electrons off of our reduced electron carrier NADPH to reduce those compounds. And that gets us down to glyceraldehyde 3-phosphate, or G3P, as we've been calling it. So those are the 3 carbons that came in the form of CO2, now are in the form of an organic carbon molecule that can leave this cycle. So we've made one G3P, 3-carbon molecule from our 3CO2. The remaining carbons stay in this cycle. So you can see the next, we had 6 times 3 carbons here. Again, that's a total of 18. And then you minus 1 or minus 3 carbons here with taking our G3P out of the cycle. And then that gets us back to 5 remaining 3-carbon molecules, which gets us back to 15 carbons total, which is what we had started with, 15 carbons total. But we're not yet into the right form because we had started with 3 5-carbon compounds. Right now we have the opposite of that. We have 5 3-carbon compounds. So how do you get from the 3 carbons to the 5 carbons? There's a lot of steps involved in that. It's much more complicated than this very simplified figure is showing it. Suffice it to say, we don't have to remember what goes on there to understand this pathway. There's something that I don't know if it's an official name for it, but it's what my biochemistry instructor, when I took a year-long biochemistry class to understand this pathway, called it the sugar shuffle. And you can do a little dance when you say that. So sugar shuffle, that's just talking about the rearrangement of this, the 5 3-carbons back into the 3 5-carbons, so that we can, again, bring in more CO2 and continue doing this pathway. So let's say we do that. We bring in 3 more CO2, and we make our way around this pathway. Let me bring my arrow here. Make our way around this pathway, and again, with those 3 CO2, we're generating another G3P molecule. That pops out. Now we have 2 G3P molecules here. Oops, sorry. Banged on a button there. We have 2 G3P molecules there, which looks like halfway through glycolysis, right? We talked about that before, how you split glucose in half, you get a G3P, you get a DHAP, the DHAP gets converted to G3P. So that's where we're sitting. We've got 2 G3P molecules. Well, if you just take all those enzymes in glycolysis and you run them backwards, we can go from our 2 G3Ps back to a G3P plus a DHAP. We can connect those back together and make some glucose. And that's basically the reactions of gluconeogenesis, is just running glycolysis, the first half of glycolysis, running that backwards. And so a better way to say running it backwards is, that's backwards from what we previously learned, we're going to be running it in the anabolic direction. So that's where we take 2 3-carbon molecules, 2 times 3-carbon molecules, and we're going to make them into 1 6-carbon molecule. All right, so that finishes up the synthesis part of photosynthesis, again, done by photoautotrophs, and tells you how those plants, when they get the sunlight they need, they can make a lot of ATP and break down some water to steal some electrons from that, generating oxygen, and then that at that time or later in the day, in the evening even, they can take those products of the photophosphorylation reaction, bring in some CO2 into the leaves, combine that CO2 all together and make some sugar so that they can make different compounds the plant needs or supply you with your food when you go out and eat that little lettuce plant.